This is a production of Cornell University.
All right, thanks so much, Susan. I really appreciate it. Thanks for everybody for joining today. Super excited to kind of be back at Cornell, even though it's virtually online. So yeah, as Susan said, I'm going to be talking about this really interesting species, Northern American wild rice, or as some may know it, Zizania palustris. So just a little bit about where I'm from here in Minnesota. I'm at the University of Minnesota in the Department of Agronomy and Plant Genetics. We have about 30 faculty members. We focus on a wide array of topics related to agriculture. And we also have a pretty vibrant graduate student and undergraduate community here. One thing that's really wonderful about working at the University of Minnesota is we have these research and outreach centers that are located across the state. So right down here is where the Twin Cities is, and I'm in St. Paul. And then up here at the North Central Research and Outreach Center in Grand Rapids, Minnesota is where I have my breeding plots. We also have something called the Forever Green Initiative that some of you may have be aware of. It's a pretty great initiative that a lot of us faculty members are a part of. And that's basically where we're trying to get green cover on our agricultural landscapes year round. So we're working with perennial crops and a lot of cover crops to try to reduce our soil erosion, our nutrient runoff, and a lot of the subsequent environmental problems that can cause, especially as nutrients flow down the Mississippi River and into the Gulf of Mexico. We also have just established a plant breeding center here. It's led by Dr. Rex Bernardo, and I just wanted to let you guys know about this in case you're not aware of it. We have seminar series. We have different summer courses that we have similar to what UC Davis does, some online seminar courses. We do have a Bayer Visiting Scholar program, so if you know of anyone, please recommend them or tell them about this. It would be great to get somebody here. We haven't gotten one a scholar yet, but we're looking forward to our first visiting scholar. So let me just jump into this about northern wild rice. As I mentioned, the species is Zizania palustris. It goes by a lot of different names. So you might see wild rice as one word or two word in the literature. You might also see American wild rice, North American wild rice. Manumin is the name for wild rice, the Anishinaabe or Ojibwe word for wild rice, as well as Sin is for, comes from the Dakota indigenous peoples language. So this is an annual wind pollinated, it's a grass, it's an aquatic grass. So you can see here it has this typical grass structure. The thing that's pretty unique about wild rice is that on this panicle, we have the male florets that are located at the bottom of the panicle and the female florets where we get our grain up at the top. This species is diploid. Thank goodness. And it is a part of the Arizae tribe in Poaceae. So this species, especially here in Minnesota, it has a lot of ecological, cultural, and agricultural importance. And so I'm just going to go through a brief history for the species. So if we think about the evolution and the ecology of wild rice, specifically, let's just start talking about the genus Zizania. There's 4 species. Here is a map of Zizania aquatica, which grows within New York and along the East Coast. The species that I work with, which is Zizania palustris, is here, and you can see the range upwards into Canada. You can't find. This is actually a historical map. You can't really find Zizania palustris here in the southern regions anymore. It's a species that really likes the cold, and of course, a lot of the agricultural that's present here in the Midwest has also reduced these populations. There is also another species, Zizania texana, that's in the San Marcos River Valley in Texas. This is on the endangered. This species in particular is on the endangered species list. And then there's one other species, Zizania latifolia, which is found in China, and it's actually grown commercially. They eat the stems that have a Eustalago infection. So wild rice is pretty important ecologically here in the state of Minnesota and within this region. You know, we are in the center of diversity. I just want to point that out. It provides a lot of food and shelter for a range of species. You know, we've got pictures here of the moose, the loons, very important for waterfowl. So if we think about some of the history, the cultural history of wild rice, this is very important to Indigenous communities here in Minnesota. So this is an example of the migration story of Anishinaabe, specifically here for the Fond du Lac tribe or reservation. And so what the history there is that the creator told them to go to where the food grows on the water. So Anishinaabe migrated from the East Coast, stopping at various different points along the Finger Lakes until they wound up in Minnesota. And here you can see a picture of them in their canoes with harvesting wild rice in this painting. Today, after several treaties and ceded territories, we have reservations here. There's probably about 13 between Anishinaabe and Dakota communities. So these are sovereign nations that reside within the state of Minnesota. Here's an example of how they traditionally harvest the grain on the lakes and rivers here. So they'll take these two poles. They're typically cedar. They'll use one to pull the plant into the canoe, and then the other to knock the grain. Obviously, there's shattering here within this grain. And then also since colonialism, the wild rice has become pretty important here, even to non-Indigenous communities. It's our state grain, and also important in a lot of our dishes here. You may have heard of chicken wild rice soup, but this is sort of a staple here in Minnesota. And we have a very healthy population of non-Indigenous harvesters of this grain as well. So this grain is super nutritious. It's got a lot of fiber, it's got a lot of protein and antioxidants, more so than even brown rice. And so commercial entities, a lot of food companies were really interested in starting to put wild rice, especially into their mixes with white rice. Now, wild rice that comes from the natural stands and our lakes and rivers, you can't source a consistent supply. So sometime around the 1950s, Minnesota farmers in the north of the state started experimenting in patties. Shortly thereafter, in the 1960s, this is when research began on this species, and we also identified a shattering trait. And so we were able to to develop a non-shattering cultivar, which really revolutionized the industry. It took off in the 1960s. California picked up production around in the 1980s. They have an ideal environment for growing cultivated wild rice. As you can imagine, being outside the center of diversity and origin, they don't have to deal with a lot of the pests that we have here in Minnesota. Today, it's considered a high value small commodity crop. Minnesota and California are the main producers. And in Minnesota, we produce roughly about 9,000 metric tons on 15,000 acres. So again, a pretty small crop. We're mainly growing wild rice within the northern region of the state. So this sort of gives that history as well from 1950 to 2020, but and you can see here also, we've been steadily releasing varieties within the program for the cultivated wild rice growers here in Minnesota. One thing I wanted to point out was the history of the breeders. So Dr. Antonelli had started the program, followed by Rob Stuckard. Ramey Porter was the breeder for about 25 years. He actually received his PhD from Cornell. And then I started in 2017, late 2017. And you can see here that there's this gap. So I didn't have any overlap with Dr. Porter. And also given some of the unique characteristics of wild rice, specifically its unique seed physiology, which I'll talk about in a little bit. This caused some problems, and in many ways I felt like I was starting this program from scratch. So the main goal here for us is obviously cultivar development. And if you think about some of the technologies that we have available to us as plant breeders, starting at the very basic of phenotypic recurrent selection up to genomic selection, this is where we are at currently. This is where we've been for roughly the past 60 years. So we currently use phenotypic recurrent selection. It's an open pollinated species. So our cultivars and our populations are heterozygous and heterogeneous. And so when I came into the program, my goal is to get to develop a molecular breeding component here for cultivar development. So the ultimate goal is to get here to marker assisted selection and genomic selection for this crop. Because it's open pollinated and these cultivars are heterogeneous and heterozygous, I think genomic selection is going to be excellent in terms of cultivar development. Our main focus, so we're still, if you can imagine, the breeding program started in the 60s. So we have about 60 cycles of selection here. So we're really still focusing on a lot of domestication traits. So shattering resistance is a huge one. It's probably the main thing I work on. So here's an example of the male florets and the female florets. And I just wanted to show you this video. It is of the male florets, but we know that shattering of the male florets is highly correlated with seed shatter. So this is something that we work on quite a bit. We also have a lot of late season storms here in Minnesota, which leads to a lot of lodging. So lodging resistance is something that we're focused on. And then the main pest that we have is a fungal pathogen, Bipolaris oryzae. So today, just a couple of things that I want to talk to you about with what I've been doing over the past 3-4 years is working on trying to navigate some of the unique life cycle of wild rice, as well as the unique seed physiology. So just to go over really quickly what this life cycle looks like, the seed is going to germinate underwater. It's going to germinate underwater. It's going to grow underwater for about a month, and then it's going to get to this phase called the floating leaf stage. This is when this leaf is developing a waxy cuticle, so it can float on the water. Then eventually the plant will go aerial. It will flower, and when the seed ripens in natural stands, it's going to shatter and it's going to fall back into the water, place itself in the riverbed or lake bed over winter, and then the process will start over again. I'm also going to talk about some of the genomic resources that we've been building, as well as some of the initial germplasm characterization that we've been working on as well. So just to orient you guys a little bit more again to what we do. This is how we grow wild rice. So wild rice seed is recalcitrant. So basically you can't dry it down and it can't stand low moisture content. So we have to prep everything for planting the day of, or maximum the day before. So here's us getting our seed trays ready. We will plant on dry land, just like any other upland crops. And then we have to flood the paddies immediately to make sure that seed does not dry down. We do this in paddies that have raised dikes. You can kind of see the green dikes right here. The plants will stay in the water for about 3 months. And then we start to drop the water down so we can get in to do our selections as well as to harvest the grain. And you can see here that the ground is quite dry. Once we make our selections and we harvest here with the seed, we have to collect all of our post-harvest traits within like 48 hours, because again, we can't let the seed dry down. So there's parts of the year where we're really scrambling to do as much as we can. And it's definitely a point where as many people as we can get out there is super important. So I had mentioned that wild rice is an outcrossing crop. It's specifically wind pollinated. So we are really limited spatially with these patty structures. So this is an example of a patty that's 500 feet long by 40 feet wide. And because we have these spatial limitations where we can't just grow wild rice in any field, historically, these populations have been butted up right against one another. So you can see here's one population that's 40 by 40 feet long. We know that we know that pollen-mediated gene flow is a problem. We wanted to start to characterize that more. So we have used a recessive white flower trait, male floret trait. So you can see here, this is the typical purple male floret trait. Here's the white flower. We also can look, we've identified that we can look at the leaf color as well as culm color. So you can see here the purple and the white. And so basically what we did was, we've done a couple of these studies, or this is one of them, where we have a all purple plot. And then everything else, all of these other plots, these are 10 by 20 feet. I apologize for the switch from metric from switching from the different systems. But we have this purple plot and we're evaluating how far pollen travel extends by looking at the progeny of these white flowers plants, because this is a recessive trait. And so what you can see here is that we've identified that the majority of the pollen travel is happening within the first 20 feet within our patties. So this isn't actually representative of maybe what happens in the natural stands, but at least what happens within our breeding plots. So I forgot to put a scale here, I apologize, but this is about 20% here, this color. And then as we go out into these further sections, this is about less than 3% pollen-mediated gene flow that we're identifying. So we're really having problems here within the first 20 feet. If you can imagine, if we have, say, population 2 right here in a 40 by 40 foot plot, and you go in 20 feet both ways, you literally have nothing to select from. So now we know that we need to start expanding these. We've been building more patties. And so we're trying to figure out ways to get around this pollen-mediated gene flow. So now I'm going to switch gears and talk about some of the unique challenges of the seed physiology. I think this is one of my largest challenges here within the program. So there's two main types of seed. There's orthodox seeds and there's recalcitrant. And then when you get deep into the discipline, some folks want to see this category of intermediate seed. So orthodox seed, this is going to be your wheat, your oats, your white rice, lots of your cereals that are desiccation and also cold storage tolerant. And so what that means, again, is you can dry those seeds down to a low seed moisture content and you can store them long term. Recalcitrant seed is desiccation insensitive or intolerant, and it typically has no cold tolerance. And so what this means is that long-term storage is often not possible. Some examples of this are tropical fruit trees, including avocados and rubber trees. There is this intermediate class where it's partially desiccant tolerant. There's limited to no cold tolerance. Wild rice does fall into this category because it does have cold tolerance. Wild rice wants to be in the cold, which is one reason why I love it. And so some other examples can be the papaya, the coffee bean, and the macadamia nut. Now, when we're talking about recalcitrance, this is just an example of what I mean by viability versus moisture contents. In wild rice, you can see at very low moisture contents, the viability of the seed, there's no viability, it drops off. As you get up to higher moisture contents, particularly above 30% water content in the seed, you're going to have reach your maximum viability of the seed. So if we look at this graph and we think about the time that we can spend that a seed can spend in storage versus its ability to germinate, I'm not really going to talk about vigor. Every seed, every species, right, that we store in ex situ storage is going to eventually have a specific amount of time where you can store it, and then that viability or that percent germination is going to drop off drastically. Because wild rice is recalcitrant and can't survive those low moisture contents. Another trait that is also associated with that is that limited storage time. So we find that for wild rice, we can actually only store the seed for about half a year to a year and a half tops before we start reaching this really sharp decline in viability. So what this means for my program is I have to do annual grow outs of all of my germplasm, which limits the amount of germplasm that I can have within a program. So currently how we store wild rice seed, or historically how wild rice seed has been stored ex situ, is at 1 to 3 degrees Celsius and submerged in water in the dark. And you can see some examples of this here in one of our cold storage areas. This can lead to a ton of microbial growth. I've done some characterization of the seed microbiome. It's really interesting. I wish I had time to talk about it today, but I will have a paper coming out soon on this. This has been a lot of fun, but we do have a lot of microbial growth, and we have found that actually the microbes don't really affect the viability and the longevity of wild rice in storage. So one thing that I have found, and what's fun about science is it was a complete happy accident, is I got like a 50-pound bag of seed from a grower during the planting season, I think of 2018, because I needed some seed. And he gave it to us. I popped it in our cold storage. And so it was stored moist, but not wet. Now, over the winter, this had been stored on the farmer's in the farmer's irrigation ditch. That's how they store their seed actually from year to year. They put it in these big bags. They throw it in their irrigation ditch. So it's submerged in water and it experienced those cold temperatures. So similar to how we store it, but obviously different. Anyway, so when I got it, one thing I had a technician that said, Jenny, I really want to test the germination of the seed every month and see how it compares to our storage, because we were trying to find a way to increase the longevity of our seed storage. And I said, yeah, sure. So this pink line is what I'm calling our one-phase seed storage, and this is typical of how we have been storing seed. Again, that 1 to 3 degrees Celsius in the dark, submerged in water. And you can see that after about a year, we have this rapid decline in viability and of percent germination, excuse me. And then this what I'm calling a two-phase storage, which is basically where it was submerged in water for about 9 months over the winter, or 8 months, excuse me, over the winter. And then it's just stored moist in cold storage at 1 to 3 degrees C. You can see that we're maintaining viability, and this is after about 2.5 years. So for my program, this has been a huge discovery. We're doing more studies right now, and it seems Like, we have found a way to store wild rice seed at least for 2 to 3 years, which I know doesn't seem like much, but when you do annual grow outs of all of your germplasm, the idea of maybe being able to cut that in half is huge for our plant breeding program. Oh, and there that is. Another thing about wild rice seed is it's dormant. Oh, I apologize. Is it's dormant at harvest, and it takes about 3 to 6 months to stratify and break that dormancy. Previous research has shown that you need to store the seed at 1 to 3 degrees Celsius submerged in water for about 3 to 6 months. So you can see that we sort of used our previous methodology in storage also because we knew that that's what needed to be done to break dormancy in this species. Here you can see an example of the seed. One thing that's really fun, I haven't been able to work much with this yet, is this is a tetrazolium stain, which stains metabolically active tissue. You can actually, we found that you can bisect this wild rice seed, you can stain it, and then it still will germinate. So we're trying to pinpoint and identify specific parts of the seed, specifically the embryo, what needs to be stained, also looking at histology to try to understand the relationship between embryo development, seed dormancy, and germination. So this, by all means, is not a complete list at all of seed dormancy, but in wild rice, you know, you can have, or any species, you can have a physical barrier, which would be like the seed coat. You can easily get around that by scarifying to the outside of the seed coat. So like scratching it somehow mechanically, for example. There's also an embryo barrier to seed dormancy. There's two main types that I'm just going to go over as an example. You can have a dormant, fully developed embryo, and the way you can get around that and you can break that dormancy is through a combination of hormones in the environment. And then also, if a seed is dormant, you can also have at harvest the embryo might be underdeveloped. And it might need that 3 to 6 months time to actually continue to grow into a developed embryo that can germinate. Workaround, probably maybe something like selection. And I forgot to mention that the reason why seed dormancy is important to us in the plant breeding program is if it takes 3 to 6 months to break dormancy, we're limited currently right now to 1 cycle of selection per year. Which obviously reduces the efficiency and our ability for any type of faster breeding, as well as a hindrance, as you can imagine, to genomic selection as well, because we want to be able to get into the greenhouse to genotype our plants. So we've been working for about 3 years on a bunch of different types of hormonal scarification treatments, as well as different environmental treatments. We've tried gibberellic acid, ethifon, kinetin, fusicoccin, the combination of hormonal and scarification treatments, different times after harvest, and nothing has worked. So all of the literature when I came in all pointed to that wild rice had a fully developed embryo. I haven't been able to find any combination of any types of treatments that have successfully broken that dormancy at a high enough rate for the plant breeding program and turned into viable plants. So, you know, sometimes science doesn't work the way you want it to, or at least your results. I've sort of thrown in the towel right now in terms of trying to find a way to hormonally or chemically break dormancy in wild rice. And we're going back to the drawing board, and we're asking the question, is the wild rice embryo actually fully developed? So some of the work I'd mentioned that we're working on histology and some tetrazolium staining to try to do that. Another thing that we've been working on is we never had a dormancy curve in wild rice. So we've established one. So these are 3 different genotypes. This is weeks in storage on the x-axis, as well as percent germination on the y. And so what you can see here is weeks in storage at 0, time point 0, this would be like at harvest. So you can see this, that most of the wild rice seed is dormant right at harvest. And then as we get further into the stratification period, somewhere between 16 and 20 weeks is when the majority of wild rice seed is going to completely break dormancy. We do have a natural stand in here because I was questioning whether breeding through some sort of indirect selection or genetic bottleneck, we had had some problems with breaking dormancy. We had maybe somehow extended it. It seems pretty similar. We may have extended dormancy a little bit. So one of the things that we're doing right now is that at each one of these time points for each of the 3 genotypes, we have extracted RNA from the seeds and ran some RNA-seq to try to get it to some of the metabolic pathways, potentially, you know, genes of interest. A lot of genes in seed dormancy have been well characterized, especially in white rice. So we're hoping for that. Another thing that I look at here on this graph is potentially an opportunity for selection to reduce the length of seed dormancy. Because if you can imagine here, we do have some seed breaking dormancy, but we also at these later stages aren't seeing a huge drop in viability. I know that we are seeing some here, but what that means is that the seed, so these are all individual bags. And so what that means is that, you know, when you're looking at percent germination here that says at about 70%, you do have a percentage of those seeds within that bag that did break dormancy here early on, but are still maintaining their viability. So I think that there are some good selection opportunities here to reduce seed dormancy in wild rice. Just some final comments on wild rice physiology. We are not seed physiologists. So when I came into this position as a new plant breeder, I did not realize how important seed physiology was and how it dictates so much in wild rice breeding, in the breeding process. And so we are sort of boots on the ground learning as we go, but I think we're making some progress. This unique physiology does impose these significant challenges to the breeding program. Specifically with recalcitrants, we have to grow all of our germplasm out every year. And specifically with dormancy, we can only currently get 1 cycle of selection per year. Timing of these experiments is really difficult for storage testing or storage longevity testing. It takes years before you can get your first meaningful data point. And a lot can happen within those years. Coolers can break down, et cetera. And dormancy testing, this needs to be prepared right at harvest, which I mentioned when we're trying to get seed collected and all those post-harvest traits. It's a really crazy time in the program. So this can be a challenge as well. So, you know, I think progress I say slow, but I've only been here for 3 years, so maybe I am making some good progress. I'm going to give myself a little pat on the back. But, you know, we have identified this new storage methodology, which seems to extend longevity for wild rice ex situ. And let me just mention that wild rice, one of the big problems when we think about conservation is that it's not stored in any seed banks because of the limited longevity in storage. So starting to understand more of this relationship between recalcitrants and storage longevity not only helps our breeding program, but it also helps the conservation when we're talking about ex situ storage of all of the genetic diversity that's out there. And then again, I think that moving towards selections for reducing dormancy might be the way that we're going to have to go. So I'm going to switch gears and start talking about genomic resources. When I came here to the university, I was charged with starting a molecular-based breeding program, but we didn't really have any genomic resources. There had been one or two, like two studies, two QTL mapping studies with AFLPs and one with SSRs that had been identified in white rice and then utilized in wild rice. And I've worked on that in a couple different species, and those transferring SSRs from one species to another, in my opinion, can be really problematic. So the first thing I set out to do was sequence the genome. We have sequenced, assembled, and annotated the Zizania palustris genome. This was done, the sequencing and assembly was done by Dovetail Genomics. It's a company out in California. And then both the Genomics Center and the Minnesota Supercomputing Institute helped in this project as well. And this couldn't have been done, obviously, without this wonderful team of postdocs that I worked with. Excuse me. So a little bit about the assembly. For those of you who like these types of statistics, I like to focus on the N90 and the L90. So total length. Well, excuse me. Let me take a step back. We didn't just sequence. We used PacBio sequencing. We also generated both Chicago and HiRISE libraries for this assembly, and we use Dovetail's proprietary HiRISE assembly software to assemble all of these contigs. So when we look at N90, we're at about 39 megabases, L90 at 14 scaffolds. If you remember, wild rice is a diploid with a haploid chromosome number of 15. So I feel pretty good about this L90 number. Here's an example or a figure of the number of scaffolds we had. In total, about 2,100. And then here are our 15 largest in terms of scaffold size that you can see here. So again, pretty happy with some of the assembly that we have of some of these larger chromosomes or pseudomolecules. Busco scores. So this is going to tell us sort of how well we did in our gene space, in assembling our gene space. So 89.4%, it could be higher, but it's not too bad. I'm pretty happy with it for what we're going to use the reference genome for. LAI scores, so this kind of tells you how well your repetitive regions are assembled. Anything under 10 is considered a draft genome assembly. So we don't really have very good assembly of our repetitive regions. So just a little bit about annotation. Here you can see that, I can totally see how some people might have a problem with this. I named the chromosomes. No one really cares about wild rice, so I felt I could do it and get away with it, and I did. Most folks probably these would be called pseudomolecules, but so we have our first 15 chromosomes. We also have a couple larger scaffolds left over, which is pretty common when you have a de novo assembly. In terms of structural annotation, the blue is our repetitive regions. The purple is our gene space. So we now have an idea of where these different spaces are located in the genome. We also utilized evidence-based annotation, where we had 8 different tissue types that can be seen here. And we ran RNA-seq on them. And so now we have about 46,000 protein coding genes, as well as we've identified shared orthologs. And this is just an example of where we looked at some disease resistance genes. After working in Peter Balland-Curdy's lab, I'm super interested in innate immunity. So we have some pattern recognition receptors and similar things like that. And you can see where they're clustering in these genic spaces. So this is probably my favorite part of the talk. which is the comparative genomics section. So here is a macrocollinearity plot. Here's the 12 chromosomes of Oryza sativa on the bottom and the 15 of Zizania palustris on the top. And what these gray bands in between show is just this, the synteny, the connection between the two species. And so you can see large overlap or large inversions, large duplications. If we look at a microcollinearity plot, so this is where every block is a different gene. You can see that there's pretty good microcollinearity on a gene by gene basis as well. One of the coolest things that we identified, which might be kind of hard to see, it's easier to see in the dot plot in the paper, is we identified a whole genome duplication in Zizania palustris. This was hypothesized in some of the work that previously been done on the Zizania latifolia genome. So I guess I should, rather than say we confirmed that a whole genome duplication happened within Zizania. That's more appropriate to say. So when we compare some genomic statistics, we have Oryza sativa here. Zizania latifolia does have a reference genome assembly. It's a bit more broken up than ours, so the scaffolds are much smaller, but we still were able to compare it. And then Zizania palustris, which is ours. So in Oryza sativa, the genome size is 370 to 430 megabases. Zizania latifolia 604. So you can almost see that whole genome duplication here. And then with wild rice, the genome's huge. So this is our assembly, it's 1.2 gigabases. Based on flow cytometry, the size of the genome is actually 1.8 gigabases, much larger than we expected. And we really questioned at the beginning of this whether or not we had another whole genome duplication after Zizania latifolia and Zizania palustris split. When we started looking at comparing the number of genes as well as the percent of repetitive regions, one thing that you can see here is that between Oryza sativa and Zizania latifolia, excuse me, percent of repetitive regions are pretty much the same, and we have this huge jump between Zizania latifolia and Zizania palustris. So our current hypothesis is that we had a rapid expansion of repetitive elements within the wild rice genome after Zizania latifolia split and Zizania latifolia and Zizania palustris split. And it's happened within, we know that we hypothesize that it's happened within the last 5 million years. So this is really similar to the evolutionary history of maize. And so if we look at a dendrogram, just to sort of orient you a little bit more, here is wheat, here is maize, here is the Oryzaceae tribe, okay, here's Oryza sativa, here's the Zizania genus. We had already known that Oryza and Zizania split from one another about 25 to 30 million years ago. We confirmed that again with our work here with this genome. But one thing that was new is we identified that this split between palustris and latifolia probably happened between 6 and 8 million years ago. One thing that's really interesting, and I don't quite understand it, molecular clocks aren't perfect, is that we know the whole genome duplication in the Zizania genre happened, we estimate 5.3 million years ago. So it seems like there's something that happened when the species split, as well as the whole genome duplication. We haven't really been able to unravel that yet. And so now that we have this genome, this reference genome, I am super interested in the domestication syndrome. I think it's fascinating. The idea that, there's lots of different traits within a wide range of agricultural species. that have this convergent evolution where some of the same genes have been selected for example, reduce seed shatter. I just love this work. This is sort of something I've really excited to begin working on. So we really want to use comparative genomics to look at the domestication syndrome. As I mentioned, seed shattering is really important for developing new cultivars and wild rice. This is an example. This is a paper from white rice, and this shows you Nipambare, which is shattering resistant, so it doesn't have the excision layer, right? And so this is a near isogenic line that shows with the gene QSH1 or SH1, excuse me, that has that excision layer. So this is the point between the grain and the pedestal where you're going to have that excision layer. And if you have that layer, the seed is going to shatter. And so I hope to get a good picture of like this of cultivated wild rice. We've got a few. They don't look at this as nice as this. So I kept this in here, but we can actually, we've started to look at the excision layer through histology. We've also identified some orthologs between some of the major shattering resistant genes or shattering associated genes in mainly Oryza sativa, and we've looked at and mapped them into the wild rice genome. One thing that I'm super interested in is because we had that whole genome duplication, how is that affecting? All right, so we have these genes that have been duplicated, and so how is that affecting their path? So typically, or their evolutionary history. So typically, one gene will remain selected on, and the other genes are free to change. And so typically they will be silenced or turned off. Other times they mutate and they develop new functions. So that's something I'm super interested in as well. I'm also really curious, we're working on it right now, how is this whole genome duplication going to affect like QTL mapping, the development of linkage maps, positions and of SNPs and such. Anyway, sorry, I digress. But so we've identified these genes. I have a PhD student right now, Reneth Milas, who's working on RNA isolation and RT-qPCR. So she's looking at genotypes that are more shattering resistant in wild rice, and also genotypes that are more shattering prone. And so we're going to analyze those gene expressions and hopefully get to some genes that we're interested in. Remember, I'm not quite at QTL mapping yet. We just started developing some linkage maps because there's other work here that I wanted to do first. And so that gets to establishing a genotyping platform. Sorry about that. So SNP chips, obviously, it's a way I would love to go, but they're possible. I can piggyback off of a white rice SNP chip, which I might do someday. It might be possible, but they're obviously costly. And I don't have a community. There's one other wild rice breeder in the country, Charlie Brummer at UC Davis, and it's kind of a side project for him. So we went with genotyping by sequencing. Some of the early, and we based our, I don't know what's going on with my apologies. We based our first initial restriction enzyme selection on early genome size estimates, which were about 600 megabases. So we did this pilot study where we looked at number of reads per sample and the number of polymorphic markers that we got from that. Once we had the reference genome and we did the flow cytometry and learned that, okay, the size of the wild rice genome is actually much larger at 1.8. We went back and we did an in silico digestion of the genome, and we found that this set is was much better. And you can see when we look at 2 million reads per sample, we go from 2,500 to 37,000. So, this is an easy study, but I think it's just as good to highlight sometimes when you're working with a new species or a new technology, you really have to do your due diligence to look at the method, to optimize the methodology. Because this obviously at the same cost of 2 million reads per sample, we jumped up about 35,000 SNPs. So huge improvement for us. And I just want to end with some of the genetic diversity assessments that we've been doing. We have collected 48 samples from about 13 different lakes distributed across northern Minnesota. We have Zizania aquatica as an outgroup. This little star is the Nick Rock. This is where the breeding program is. These highlighted counties are where we grow cultivated wild rice. And if you look at this PCA plot, so if you're not familiar with a PCA plot, what this is basically every dot is an individual, and the colors correspond to different lakes. So you can see that there is structure. We've identified some structure based on watersheds, geographical locations, which hadn't really been established before. Here's Zizania aquatica. If you look at different PCA principal components, this will break apart from the natural stands. But most importantly, here's our cultivated wild rice germplasm, so that you can see that even just with 60 cycles of selection over the past 60 years, we have been moving and selecting to get wild rice is distinct from the natural stands. We're now trying to understand gene flow and have some questions about that, about similarities between these two groupings. This is a big thing because we're growing cultivated wild rice and we're changing some of the genome of this species to adapt to these agricultural climates, but we're in the center of diversity. So we have to be really careful. We don't do any mutation breeding or genome editing in my program. There's actually a law in Minnesota where you can't develop any GM wild rice because folks were really worried about that about 15 years ago. You can't develop that without a environmental impact study. But there's validity there because we're in this center of origin. We don't want to disrupt the natural stands at all by breeding and cultivating the species. And so I was really happy to see this. It appears that there's limited gene flow between the cultivated stands and the natural stands. We've been starting to preliminary look at LD decay. This is a outcrossing species. So LD decay is pretty rapid. It's similar to what we're seeing in maize. This is a dendrogram of everything in like maybe 15 different cultivars or populations within my cultivated wild rice breeding program. I know you can't see it, but if you can see the different colors, there's no structure here. The only structure that we see is with K2, which is a population that's been physically isolated in a whole nother paddy that's far away from the main paddy complex for the past 30, 40 years. So I think because of all the pollen travel that we know we see with the high level of LD or, the rapid decay of LD, this is contributing to just this mess that I have in the breeding program. So trying to implement and institute some structure within our breeding populations, reduce that pollen travel is definitely a main goal that I'm working on. And lastly, and I love this, is a PCA of 2 different lakes. And this is towards some of the conservation goals that I have trying to help within the program. So here's Garfield Lake. The red dots are from samples collected in 2010. The black dots are from 2018. So you don't see a lot of diversity or population structure change. Here is another lake. The blue dots are from samples collected from Shell Lake in 2010. And then the green dots are from the same lake, Shell Lake, collected in 2018. And you can see that there's been this rapid reduction in genetic diversity. So I'm really interested in expanding this and looking and asking questions as to why. And so that means using maybe some remote sensing, some landscape genetics, land use modeling. And I can say anecdotally, just anecdotally, that Garfield Lake is a lake that's been pretty closed off, and there hasn't really been any development over the last 10 years. Shell Lake has a lot of recreational activity on it, and the surrounding areas have been rapidly expanding. So there's some anecdotal evidence that we are harming and reducing the genetic diversity. And for an outcrossing species, making sure that you have a very diverse populations is incredibly important for their fitness. So I'm hoping to expand this, do a little bit more research in this area, and hopefully try to come up with something that might help conservationists and in some of their management strategies. So that's all I have. Thanks so much. It looks like I only went 5 minutes over, so I'm pretty happy about that. Yeah, this is the team. Collaborations aren't really inherent in wild rice because there's not a large group of us working on wild rice. So seed physiologists, landscape, modeling specialists, anything. If anyone's ever interested in working on wild rice or knows of someone who will or who might be interested, I think that's fantastic, and I'm always open. And so with that, I'll take any questions. This is just a picture of our paddy complex in upstate Minnesota. So thank you. Okay, I'll start in the chat and look at these questions. Is that okay?
Yep.
Okay. So since wild rice is immensely important to the Ojibwe Nation, are you working with them to provide opportunities to Native students for mentoring and plant breeding? That's a great question. So I don't have any Native students. I would love to. There's definitely a stigma. I don't think a Native student would be really well received within their communities if they knew that they were working on wild rice, honestly, especially cultivated wild rice. There's a huge history between the university and these tribal nations. We do have one thing that is super interesting is we do work with Red Lake Nation. They actually grow cultivated wild rice. And so that is where I see opportunities right now to sort of break through some of those barriers and get some students. But I would love that. I think that would be absolutely wonderful. And even if they weren't working on cultivated wild rice, just to work on the natural stands and some of those conservation aspects that I would like to, I think that would be great. So the next question is, have you had discussion with the Indigenous community about wild rice improvement? Are they interested in wild rice improvement and the possible impacts on their use of wild rice? It seems like this could produce a very interesting case study where breeding seeks to promote equity. So yeah, kind of along those same lines that I was mentioning. So one thing that's really important, right, with any community is to understand that they're not a monolith. So we do work with Red Lake First Nation, which we have a very good relationship with them. There's other First Nations in Minnesota that really don't want us working on wild rice. And so that's, it's a challenging space to work in as a Western scientist. In graduate school, we're not really taught how to navigate these situations. I will say that when I was at Cornell, I took a class, and I don't know if it's still there. Hugh Gao was someone who was one of the instructors, and it was called Putting the PhD Back into PhD. And it talked more about the philosophy. And I think that class, more than anything else that I took, has helped me sort of navigate some of these situations. So I really want to be an advocate. I want to be someone who helps these communities. I have a long way to go to do that for sure. There's hostilities and barriers in place that have been developing for hundreds of years. I've only been here for 3 or 4 years, so I'm certainly not going to break through those barriers immediately. But I hope if I can, through my research, show some interesting and novel concepts about conservation that some folks can maybe try to at least come to the table and have some conversations. It's challenging, but I totally remain hopeful. As an open pollinated heterozygous species, how are the wild rice cultivar identities maintained by seeds? Such a good question. They're not really. I had mentioned it's sort of like the wild west. It feels that way. There are cultivars that were released in 2014 that even when I came here in 2017, I didn't have. They had already been lost. And so there's not a seed industry in cultivated wild rice since it's such a small commodity. The growers actually maintain these seeds, these cultivars in foundation fields on their farms. And they have different parameters of, size of the field, distance to other paddies, etc. So yeah, and that is something that they really want. They really want to be able to have like molecular markers that can identify these cultivars. Are you receiving support for your outreach efforts in terms of your tenure at the UMN? That's a really good question. No, not so much. This is a problem. This is an issue that, as I mentioned, has been hundreds of years in the making. The university in general has struggled. The college that I'm in has struggled. The department that I'm in has struggled, and no one has really come up with effective solutions. I will say that we have a new president who's been doing fantastic. We have new tribal liaisons. And we also just yesterday, it was announced that we are providing free tuition to all Native American students from the tribal nations here in Minnesota. Gets free tuition. So there's a lot of things at work and at play here in Minnesota, I think, to try to bring up this marginalized community. We also are Lieutenant Governor Peggy Flanagan, excuse me, is Anishinaabe. So there's tensions, there's challenging conversations, but I'm really pretty proud of what the university has been starting to do and what the state of Minnesota is trying to do for its Native communities.
Do we have any questions from the audience here? Okay, there's one question.
Is that part of like a diversified operation, or are they specialized just in growing flowers?
Okay, so could you hear the question?
No, could you repeat?
It was about do growers also do other commodity crops or just the wild rice? Did I get your question?
Yeah. So wild rice is super interesting. These guys, what they'll do is they'll go into a field that's been fallow of wild rice specifically for at least 4 years. They'll plant wild rice. They're still shattering in any cultivar that we currently have. When they go to harvest, that seed is going to shatter, and they actually use that shattering seed for next year's crop. And they do that for 3 or 4 years. So it's sort of this conundrum where they want seed shattering resistant cultivars, but they also, the system that they're using selects for more shattering. But then they'll do that for about, it depends on the grower, 3 to 5 years. And then they will grow, it depends on the region, lots of soybean, some wheat, some spring peas, And then in the Red River Valley in northwestern Minnesota, potatoes. We grow a ton of potatoes here in Minnesota. So yeah, they have a rotational crop.
I have a quick question about how it is for you starting up basically from scratch. Was it a freeing endeavor, or did it actually, make things harder for you to have to kind of restart a whole program?
Yeah, it was pretty rough the first few years. The seed needs to be handled in a very delicate way, and because of the recalcitrance. And so when I came in, all of the germplasm, I think the germination rates were about 20%. So it took me 2 field seasons to get the just to the germplasm to the point where I could get 70% germination or viability. So that really was a hindrance, but I'm super blessed as a public plant breeder because I have a legislative appropriation, an annual legislative appropriation. And it had started a few years before I came here. So I came in with a pretty healthy budget. And that's why I was able to sort of hit the ground running with a lot of the developing a lot of the genomic resources. So some parts were hard, some parts were a lot easier.
I think we have one more question in the audience. Go ahead.
How are you? I was wondering, you said the genome is duplicated, and I'm wondering, you said that one gene is always conserved and the other is free to do what it wants. Is there one set genome, or does one or the other, can you tell at this point if one set of them always stays and the other always mutates, or is it just randomly which one does?
Yeah, I'm just basing that off of literature from other crops.
Okay.
But I'm so interested. I mean, that's something that we're looking at right now, is to try to evaluate, we have all of these duplicated genes in the genome, what's going on with them. We don't know yet, but it's going to be an exciting part of research. So I love this species. I love working on it. There's so many questions to ask. And never thought that I would find what I did with the sequencing of the genome, and that's just been another really fun place to work.
Well, thank you very much for your talk. It was very exciting. Can we give her one last round of applause, please? Thank you.
Thank you, everyone. It's a joy and pleasure.
This has been a production of Cornell University, on the web at cornell.edu.